Hi everyone, today we're going to be solving AQA GCSE Biology Higher Tier Paper 1H June 2021 question paper. The first question is about microscope. A student prepared some animals to view using a microscope. Figure 1 shows the student preparing the cells. We can see the microscopic slide where the student is placing a cover slip on a sample. Name two pieces of laboratory equipment that the student could have used to prepare cells to view using a microscope. So we can name microscope slide, cover slip, which is already shown in the picture, but other than that the student may have used dye, the student may have used mounting needle, then the student may have used a dropper or a pipette to put the dye in that particular place, the student may have used a scalpel to cut that particular sample, or a forceps so that the student can place that particular sample in the or a forceps so that the student can hold that particular sample while cutting it with scalpel. All of these are possible answers. You just need to write any one of those two, but my personal preference is the one that is shown in the picture, which is microscopic slide and cover slip. Figure 2 shows the student's light microscope, and the part A and part B, they're asking the name for part A. So we can see the part A is the place where the student places his or her eyes to see it, so we call it eyepiece lens. Now the next question says, what is the function of part B? The part B is actually adjustment. So it is used to focus the image. The student tried to look at the cells using the microscope. Suggest one reason why the student could not see any cells when looking through part A. There can be many possible answers for this particular question, and this particular question is very common whenever a microscopic question is asked. First point would be no cells in the field of view when the student was viewing through the eyepiece lens. The other points are the slide is not in the correct position, or the mirror is not in the correct position, or the objective lens did not click into its place, or the student is looking at a large air bubble, or the microscope is not focused. You can choose any one of this particular point from the answers that I am providing. However, my personal best from here is that either you can say no cells in the field of view, or the objective lens not clicked into place. Red blood cells are specialized animal cells. Compare the structure of a red blood cell with the structure of plant cell. This is a 6-mark question, so to get a level 2 response, to get the 6 marks, we need scientifically relevant features, we need to identify them, and then the ways in which they are similar or different, we need to make it very clear, and where appropriate, the magnitude of the similarity and difference, we need to note it down. Because the question says about comparison between red blood cell and the structure of a plant cell, so every time we need to write for both of them. It is a better idea to write for both of them because it is a comparison. In the case of a difference, we can write only for one of them as a point. So what can we write as an answer for this particular question so that we get a 6 mark? First of all, we can say red blood cells have no nucleus, whereas plant cells have nucleus. The next point we can write, RBCs have no cell wall, plant cells have cell wall. Red blood cells is biconcave disc structure, whereas plant cells can have different shapes. The next point can be written as RBCs contain hemoglobin, plants does not contain hemoglobin. The next point can be written, red blood cells do not contain chlorophyll, however plant cells may contain chlorophyll. Red blood cells has no chloroplast, plant cells have chloroplast. And finally we can also write, red blood cells have no permanent vacuole, plant cells has permanent vacuole. Red blood cells are much smaller than plant cells. Now guys, we also need to write some points for similarities. So for similarities, we can say that both have cytoplasm, cell membrane, and pigments, however they are different. Since this is a particular comparison question, so it can be written as the way written in the beginning, or it can be structured totally in a format of a table. You can use any one of that process, whichever is convenient for you. So I have shown you half and half, alright. However, you do not do half and half in your exam. Rather, you choose either this method or the method that I have used at above. And then finally, when writing similarities, you can write it like a statement, because however the similarities are like a statement that they both contain this. When placed in beaker of water, a red blood cell bursts. A plant cell do not burst. Explain why red blood cells burst, but the plant cell does not burst. Water enters the plant cell or the animal cell by osmosis. However, the plant cell has a cell wall which prevents it from bursting, and red blood cell does not have cell wall, so it swells and bursts. A student investigated the effectiveness of 3 different antibiotics. Figure 3 shows how the student set up an agar plate. We can see the bacteria growing on agar, and we can see filter paper containing antibiotic A, B, and antibiotic C. The student used aseptic techniques to make sure that only one type of bacterium was growing on the agar. Now, obviously, whenever a aseptic technique question, is mentioned, the next point will be describe 2 septic techniques the student should have used. So septic techniques involve the use of sterilization. So the possible answers could be the student used sterilized equipment before use, the student used sterilized agar. These are the best possible answers. And then some alternative answers will be the student used the lid of the petri dish was secured with adhesive tape so that only one type of bacteria grows, and the lid of the petri dish was lifted a little when setting up the plate so that no new bacteria is introduced within that particular agar plate. So those 2 points are alternative points, and the first 2 points are the better points. You may choose to write the first 2 points. The student placed the agar plate in an incubator at 25 degrees Celsius for 48 hours. Figure 4 shows the agar plate after 48 hours. Now we can see some clear regions. Those clear regions, the radius of the clear region will indicate the effectiveness of that antibiotic. So we can see C has the greatest clear region, whereas B has the lowest clear region. Which antibiotic is the least effective? And give a reason for your answer. So the least effective antibiotic is B because it has the least clear region around it. Or an alternative answer for this one will be it kills the fewest bacteria. 2.3. Calculate the area where no bacteria were growing for antibiotic C and use the formula pi is equals to 3.14. To do this particular method, we will have to calculate the radius for C. Now, in the tablet, it's not possible, so we're using from the mark scheme. The clear region is 1.1 cm, which is the radius for the C. So first of all, we will use a ruler to find that out. So R is equals to 1.1 cm. Then we will use the area formula. The area for a circle will be pi r squared. Recalling this one will give us one more mark. Then we're going to use the pi value 3.14 times r square, which is 1.1 whole square, and this will give us a roughly 3.799 cm square. Now we will mention the unit, because our question may ask us sometimes to give the answer in standard form, so we will think of the answer in the standard form. So area is 3.799 and the unit will be cm square. We may also some of you may also opt to write out 3.8 and still be correct. Suggest one way the student could improve the investigation. One way of improving any investigation is to repeat and calculate average or mean. The other ways of improvement for repeating this particular experiment would be using a control disk or use a different type of bacteria. Body mass index BMI is a way of finding out if a person's body falls within a healthy range for their diet. Table 1 shows the information about 2 people. We can see person A who has a body mass of 63 kg, a height of 1.65 and BMI of 23.1. Person B has a body mass of 92 kg, 1.71 is his height and the BMI is unknown. So we will need to find X. The question says which is the BMI category of person A in table 1. So for person A, it's 23.1. If we look into 23.1, then we will find out 63 and 1.65. So first of all, we will go 1.65, which is here, and then we will go to the mass of 63, which is right around here. 63, which is the mass is right around here. So this particular person is normal. Now the question says calculate the value X in table 1. Now to calculate BMI, we use the formula BMI is equals to body mass divided by the height squared. So to calculate BMI, we are going to write the body mass, which in this case is 92 kilograms divided by 1.71 whole squared. So our answer is 31.46. Scientists think there is a link between BMI and life expectancy. Table 2 shows information about the predicted life expectancy of men after the age of 50, with normal age predicted number of years living in good health after the age of 50 is 19.06, and with bad health predicted number of years to live is 4.98. Now, as the person becomes overweight, obese, and clinically obese, the living with good health decreases significantly from 19 to 13. However, as the person's BMI increases, all right, living with bad health after the age of 50 increases significantly to 10.10. Describe two patterns shown in table 2 about the effects of BMI category. So first of all, we can say the higher the BMI category, the lower the number of years living in good health. Now, the other conclusions that we can make, the higher the BMI category, the higher the number of years living in bad health. An alternative answer will be the higher the BMI category, the lower the total life expectancy. The number of people who are obese in the UK is increasing. Explain the financial impact on UK economy of increasing number of people who are obese. Since the person who are going to be obese, they are going to be more sick, it will cost the NHS more money because the UK Health Services System or the NHS need to pay more money for medication and hospital stay to treat stroke, surgery or diabetes. So the employer or the government have to give more financial support to the employee. Now, out of these 3 points, only 2 will be needed for scoring 2 marks. So you can choose. A person who is obese is more at risk of arthritis. Arthritis is a condition that damages joints. Suggest how arthritis could affect a person's lifestyle. There can be many points for answering this particular question. The most important one is that the persons will have movement issues. So this can have other leading consequences from there. The person will lose their job. The person may enter disability. The person's mental health will be impacted due to lack of movement. The person may need to visit the doctor more often, and the person may need to undergo surgery. All of these are valid points. We just need only 1 point from all of this to get our mark. A person who eats a diet high in saturated fat might become obese. Name 2 health conditions that might develop if a person eats a diet high in saturated fat. Do not refer to arthritis in your answer because it is already mentioned before. So, the person who is obese can develop type 2 diabetes, cardiovascular disease, may have heart attack or stroke. All of these are valid points. Just write 2. All living organisms respire. What is the chemical equation for the aerobic respiration? So aerobic respiration involves the breakdown of glucose molecule. So only 2 of the equation consist of glucose molecule. So we will reject the other 2 because the other 2 does not contain glucose as a reactant. And only the last equation consists of oxygen that is used with glucose. So oxygen and glucose produces water and carbon dioxide. Water cannot break down glucose by itself. So 4.1, the last one will be our correct answer. Name the subcellular structure where aerobic respiration takes place. Aerobic respiration takes place in mitochondria. Guys, as an interesting fact, mitochondria is actually an early bacteria that came to live in eukaryotic cells in a symbiotic relationship, and then it is ever since present in eukaryotic cells. Energy is released in respiration. Give 2 uses of energy released in respiration. Now, this answer can have many points as answer, but general understanding would be movement or muscle contraction and active transport, a building of larger molecules, and there can be many more examples, all referring to chemical reactions that is referred to as metabolism, but metabolism by itself cannot be the answer. Now alternative answers will be keeping warm and or building larger molecules. You can write the name of the process of building larger molecules. For example, making proteins from amino acid, or making lipids from fatty acids and glycerol, or making glycogen from glucose. You can write that instead of writing building larger molecules, and it will still be a good answer. Describe 2 differences between aerobic and anaerobic respiration in humans, and do not refer to oxygen in our answer. So, difference between aerobic and anaerobic respiration is very simple. Anaerobic respiration produces lactic acid and aerobic does not produce lactic acid. The other point that we can write is that aerobic produces carbon dioxide and anaerobic does not produce carbon dioxide. We can use CO2 in this case. You may choose to write the carbon dioxide full thing. Some alternative answers also given here. So aerobic produces water, anaerobic does not produce water. Another alternative answer will be aerobic occurs mainly in mitochondria, anaerobic does not. A final alternative answer for this one would be anaerobic releases less energy than aerobic respiration. What are the two products of anaerobic respiration in plant cells? So in plant cells, when anaerobic respiration takes place, it produces carbon dioxide along with ethanol. Glucose is the reactant, lactic acid is produced in anaerobic respiration in animal cells, and water is produced when glucose is broken down in aerobic respiration. So that's why carbon dioxide and ethanol is the correct answer. So in plant and fungi, when glucose is broken down in general in anaerobic respiration, the product will be ethanol and carbon dioxide. So it's 2 ethanol and 2 CO2. A scientist investigated respiration and photosynthesis using some pondweed and a pond snail. Figure 6 shows the apparatus that is used. We can see a data logger, a carbon dioxide probe is used in pondweed, boiling tube containing pond water with a pond snail in it. The apparatus was left in a well-lit room for 5 days. The data logger recorded the concentration of carbon dioxide continuously. After 5 days, the scientists completely covered the boiling tube with black paper. The data logger continued to record the concentration of carbon dioxide in the water. Figure 7 shows the concentration of carbon dioxide inside the boiling tube over 15 days. So, until the tube was covered in black paper, the concentration of carbon dioxide was steady up until 5th day. From 5th day till the 10th day, the carbon dioxide concentration at first gradually increases, then increases steeply, and then it stays at the same level for a certain number of days, then increases steeply back again. Explain why the concentration of carbon dioxide in the tube stayed same between day 0 to day 5. Now we know that between day 0 to day 5 the plant was able to photosynthesize because it was kept in a very good you can see it was kept in a well lit room for the 5 days, so the plant was able to photosynthesize and produce carbon dioxide and produce oxygen from the carbon dioxide. So we will answer in this way. The pondweed takes in the CO2 for photosynthesis, and the snail and the pondweed are both respiring carbon dioxide. So, the level of CO2 remain constant. Suggest why the concentration of carbon dioxide increased between day 5 and day 10. Since there was no light present, so no photosynthesis can occur. However, the snail and the plant are still respiring and are releasing carbon dioxide. On day 10, the pond snail died. On day 10, the pond snail died. Explain why the death of the pond snail caused the concentration of carbon dioxide to increase after day 10. After the death of the snail is being decayed and decomposed by bacterial action in the pond water. Therefore, the respiration of those bacteria releases more CO2 into the pond water. Amylase is an enzyme that breaks down starch. Amylase is a polymer of smaller molecules. Name the type of smaller molecule that makes amylase. Now we know that amylase is a type of carbohydrate. The amylase is a type of protein, and proteins are made from amino acids. Guys, it is very important that we look into the molecule where they are describing it as an enzyme. Remember that enzymes are protein. Now, if the name amylose is used, it's a type of carbohydrate. In that case, we would be making the amylose with glucose. Name the 3 parts of the human digestive system that produce amylase. We know that salivary glands produces amylase, pancreas produces amylase, and small intestine produces amylase. Explain how amylase breaks down starch. Answer in terms of the lock and key theory. So whenever the question is asked based on lock and key theory, it is important for us to mention who is acting as a lock and who is acting as the key, and also how the active site is functioning. So first of all, we're going to say the starch binds to the active site of the enzyme because the shape of the active site and the starch are complementary. Then we can write a chemical reaction then occurs to produce smaller molecules. Or we can say the bonds between the starch molecules are broken down to produce smaller molecules. Guys, a generalized understanding for the lock and key theory. The enzyme acts as the lock, and whereas the substrate acts as the key. A student investigated the effect of temperature on the activity of amylase. Figure 8 shows the apparatus that is used. We can see 5 cm³ of starch solution is prepared in one test tube, and 1 cm³ of amylase solution is prepared in another test tube. They were both at a 5 degrees Celsius water bath. Now, this experiment was set up as shown in figure 8. After 5 minutes, the starch solution was poured poured into the amylase solution and they were mixed. Remove 1 drop of starch solution mixture and place it in a spotting tile. Then immediately add 2 drops of iodine solution to the starch amylase mixture on the spotting tile. Record the color of the iodine solution added to the starch amylase mixture. Then the process is repeated from 3 to 5 every minute until the iodine solution becomes yellow brown or stayed yellow brown. Repeat steps from 1 to 6 using water at a different temperature and record these results. Okay, name 2 control variables the student is using in this investigation. The student is keeping the volume of the starch and the volume of the amylase solution fixed, and the student is also using 1 cm cube of amylase solution and 5 cm cube of starch solution. So the student is keeping them fixed. The other alternative points will be the time before mixing the starch and amylase solution the student kept fixed, or the student used 1 drop of mixture which the student is adding to the spotting tile, and another alternative point that is the student is keeping constant is the student is adding 2 drops of iodine solution for every every test that he is performing on that mixture. Why did the student leave the starch solution and amylase solution for 5 minutes before mixing them? The student left the starch and amylase solution for 5 minutes before mixing them to allow the solutions to reach the same temperature as that of the water. Table 3 shows the results of the investigation. Temperature from 5 to 80 degrees Celsius is used, and with 5 degrees Celsius, time taken until iodine solution stays yellow brown in minutes did not become yellow brown. Okay, so we can see that at 5 degrees Celsius the reaction did not finish. At 20 degrees Celsius it took 5 minutes, at 35 2 minutes, at 50 7 minutes, at 65 it took 14 minutes, and at 80 degrees Celsius it did not become yellow brown, meaning that the reaction could not complete. What conclusion can be made about the effect of temperature on amylase activity between 20 degrees Celsius and 65 degrees Celsius? So we can say between 20 to 65, as the temperature increases, enzyme activity increases to 35 degrees Celsius. As you can see, from 20 to 35, the enzyme activity increases. We need least amount of time. And after that, after 35 degrees Celsius, the enzyme activity decreases again. Explain the results at 5 degrees Celsius and at 80 degrees Celsius. So we can see at 5 degrees Celsius that the reaction did not take place. We can see the yellow-brown color never appeared. And at 80 degrees Celsius, yellow-brown color never appeared. So the answer can be written in this way. Iodine is not yellow-brown because starch is still present. At 5 degrees Celsius, amylase molecules have very low kinetic energy. Therefore, a pure enzyme-substrate collision can occur. Whereas at 80 degrees Celsius, the amylase has been denatured, so the starch can no longer fit the active site of the enzyme. The student investigated the effect of temperature on amylase activity. Describe how the student could extend the investigation to determine the effect of different factor on amylase activity. The student can keep the temperature constant, but then the student can change that named factor that the student is working on. For example, pH or enzyme concentration or substrate concentration, etc. Then test a range of values of that particular factor. Figure 9 shows a cross-section of a leaf. We can see the upper epidermis represented with A, B representing palisade mesophyll, and C representing spongy mesophyll, whereas the D is representing the guard cell. Which cell is most transparent? We know that the most transparent cell is upper epidermis and lower epidermis, so A will be the answer. Which cell structure in leaf mesophyll cell is not found in root hair cell? In root hair cell, you will not be finding any chloroplast. Plants lose water through their leaves. Name the cells in a leaf that control the rate of water loss. Guard cells control the opening of the stomata, and thus it can control the rate of loss of water. Water is taken in by the roots, transported up the plant and lost from the leaves. Which scientific term describes the movement of water? Here we can call it a transpiration stream. Which change would decrease the rate of water loss from the plant's leaves? Because we want to decrease the rate of water loss, and increased humidity will decrease the rate of water loss. The other points are not correct because increased light intensity increases the number of stomata that opens, thus increases the amount of water loss. Increasing density of stomata, meaning that more stomata will be open and more water vapor will be lost. Increasing temperature means water molecules will have more kinetic energy, so more water will will be lost as water vapor. So only the first option is the correct answer. Compare the structure and function of xylem tissue and phloem tissue. Now, because the question says compare, so we must write for both of them. And to get a level 2 6-mark answer, we have to write scientifically relevant feature. We will identify them. The ways in which they are similar or different, we will make it clear. And we will also write where the magnitude of the similarity or the difference, is close or is it like totally far apart from each other. We have to mention about that as well. So I'm going to give you a, indicative answer for the structure and the function for both xylem and phloem. I will mention many more points than needed and then mention to you how many points you need to write from the structure and how many points you need to write from the function. I will indicate it with color codes, so don't worry. Just go through the entire question and you will be able to understand it. So with the structure, xylem is made of dead cells and phloem is made of living cells. Phloem cells have pores in their end walls. Xylem cells do not have pores in their end walls. Xylem is hollow and does not contain cytoplasm, whereas phloem contains cytoplasm. Xylem contains lignin. Phloem does not contain lignin. Both xylem and phloems are made of cells and are tubular. Now, in terms of function, we need to write xylem transports water and phloem transports dissolved sugars. Xylem is involved in transpiration, whereas phloem is involved in translocation. And a common point for both of them would be both transports liquids or substances throughout the stem. Figure 10 shows the total volume of water lost from a plant over 6 hours. We can see the time of the day and we can see volume of water lost in cm cube. So as from 8 a.m. up until 1400, the volume of water lost in cm cube increases, remains constant for a while and then again increases. The question says determine the rate of water loss at 1200. Use the tangent in figure 10. Give your answer in cm cube per minute in standard form. So if we use the tangent, then at 1200 we see that it is 213 cm cube of water lost at 1200. We started from 8 a.m. when we had 190 cm cube of water lost. Guys, since we are asked to use the whole tangent, so we can use the entire value where this is 1500 and 230 starting from 190. So our answer will be 230 minus 190 divided by a total of 7 hours, which is 15 minus 8. This gives us 40 divided by 7, which is 5.71 cm cube per hour. Because we have to give this in terms of cm cube per minute, so we can divide 5.71 with 60, giving us the unit in cm cube per minute. Our answer expressed in standard form is 9.5 into 10 to the power of minus 2 cm cube per minute. The rate of water loss at midnight was most lower at 12. Explain why. At night, less water is lost because stomatas are almost completely closed and it's colder at night. We can also say because there is no light at night, or alternatively we can say because it's colder. The understanding of there is no light at night is that light is needed for the opening of the stomata. Since at night there is no light, the stomatas are not open, they are almost closed. Figure 11 shows where 3 of the same type of tumor were found in a patient. We can see the tumors in the lungs and in liver. This is a malignant tumor and its malignant tumors are cancers. Describe what happens to the cells when a tumor forms. In a tumor, the cells divide abnormally and uncontrollably. What evidence is there in figure 11 to suggest that the tumor in the lung is malignant? Tumor that is malignant spreads over other regions of the same person's body. So the tumor has spread to other parts of the body. Some types of cancer can cause the number of blood components in a person's body to fall to a dangerously low level. A person with one of these types of cancer may experience symptoms such as tiredness, frequent infections, bleeding that will not stop after the skin is cut. Explain how a very low number of blood components in the body can cause these symptoms. This is a 6-marker question, so a level 3 answer will require relevant point and reasons, and the causes will be identified, which will be given given in detail and logically linked to form a clear account. The three things that we can see is tiredness. Tiredness can be mainly due to lack of oxygen, so less cell respiration. So RBCs has decreased in quantity. Frequent infections can be associated with decrease in number of WBCs, and bleeding that will not stop after the skin cut is mainly is related to platelets. So if we write 2 points for each of these, then we will get the full 6 marks most likely, but we must write it in a logical sequence. So we can say tiredness is due to fewer red blood cells, so there is less hemoglobin and less oxygen transported around the body. We can also write added points like this results in less anaerobic respiration, so more anaerobic respiration takes place and less energy released for metabolic processes. Lactic acid is also produced during anaerobic respiration, which then also causes muscle fatigue. Guys, for this particular answer, I have decided that I'm going to give you at least 2 points for each of them, or 3 points, not more than that, to give you a suggestive answer, because this particular question is very common and repeats in the same manner in the exam. So instead of choosing points, all right, if I give you the points and you choose to write only them, you will definitely get a level 3, and we have to write for all 3 symptoms. We cannot skip. So I'm just gonna give you a one indicative answer for all, every time you receive a question like this. Some patients with a very low number of blood cells may be given a blood transfusion. A blood transfusion is where a patient receives blood from a donor. Different people have different blood groups. The red blood cells found in people with different blood groups, figure 12 is showing them, the antibodies that can be made by people with different blood groups. So we can see blood group A, the type antigens, and anti-B antibodies are present. With blood group B, type B antigen and anti-A antibodies are present. The blood group O with red blood cells that contains anti-A and anti-B antibodies. Antibodies can bind to antigens that have complementary shape. When antibodies bind to the antigens on red blood cells, many red blood cells begin to clump together. Each red blood cell is about 8 micrometer in diameter. Many capillaries have an internal diameter of about 10 micrometer. In one type of blood transfusion, only red blood cells from a donor are transferred to the patient. It is dangerous for a patient with blood group A to receive a blood group cells from a donor with blood group B. Explain why. So a patient is receiving, a patient with blood group A is about to receive a blood group B donor's blood. What is the problem? The main problem is anti-B antibodies in patient will bind to the anti-B antibodies in patient who has blood group A will bind to the type B antigens on the donor's red blood cells. So those red blood cells given by the donor will clump together, and the clump will be wider than the capillaries. So the cells will have reduced supply of oxygen because the capillaries will get blocked. Explain why blood group O red blood cells can be given to patients with any blood group. Both types of blood group contains no antigen on type O red blood cells, so antibodies cannot bind to the antigen of red blood cells. Table 4 shows some of the risk associated with blood transfusion. Risk allergic reaction. Probability of risk of occurring or allergic reaction is 0.9. Hepatitis B infection 1 in 3 times 10 to the power of 5. So it's like 1 in 300,000. Okay. Hepatitis C infection is 6.7 into 10 to the power of minus 7. So it's a very, very low chance. Kidney damage 1 in 70,000. Which risk has the lowest probability of occurring? So we can see having a hepatitis C infection is the lowest of occurring, so we can give that number. How did we come to that conclusion? We came to that particular conclusion with a number. All right, 6.7 10 to the power minus 7 literally means 0. There will be 6 zeros before it. All right, then 6 7. So you can see that it's a very, very low number. All right, person has a tumor blocking the tube leading from the gallbladder to the small intestine. Explain why this person would have difficulty digesting fat. Since the tube that is leading from the gallbladder to the small intestine is blocked, that means the bile will not be transferred to the small intestine. So since there is no bile that reaches the small intestine, there will be no emulsification of fat molecules. So a smaller surface area will be available for the lipase to break down the fat, and the pH of the small intestine will not be alkaline because due to the lack of the bile not being present there. bile is supposed to, turn the pH alkaline. So lipase is not at its optimum pH to break down the fat. So guys, that's the end of this particular question paper. Thank you for watching the video. Guys, best of luck for your exam. And guys, let the channel grow by subscribing and recommend it to your friends as well. I will try to put all of these question papers that I have done in a series to put it in a playlist so that you can watch them together. And guys, best of luck for your exam. Bye-bye.